So thank you so much for having me today. I have been told that you are a group that asks questions, and I love questions. So at any point in this talk, if there's something that you want to talk about, please, by all means, ask a question. Today I'm going to talk to you about the Emery 3Q29 project and the way that we have used Bionano technology to interrogate some regions of the genome that have previously been invisible to us. And I don't know if I should apologize for the fact that you're going to be humming Amazing Grace for the rest of the day, but so be it. So before we begin, I'm just going to give you a little bit of background about what 3q29 deletion syndrome is to contextualize the rest of the work for you. 3q29 deletion syndrome was first described in 2005 in just 6 patients. These individuals have a facial dysmorphology that is subtle but not striking. And I think for the people here who work in this field, the things that I'm going to describe are very common. There are a lot of genomic disorders that don't have sort of hallmark or distinguishing or characteristic features, and the 3q29 deletion falls in that category. So all of the individuals that were initially found in this study had mild to moderate intellectual disability. They had some developmental delay. Autism was a feature was diagnosed in 2 of the patients, and a third of the patients had symptoms that were consistent with autism, some social disability, but not a formal diagnosis of autism. So right away, in this initial sort of description of the phenotype, autism is a feature of the syndrome. In 2008, the definition of the syndrome was expanded. There were 14 patients that were described. And again, there was no feature that was sort of a hallmark feature. Intellectual disability was kind of present in most of the individuals, mild to moderate. Speech delay, autism, again, we see, and then a host of other features. Some individuals had heart defects, not everyone did. Some individuals had recurrent ear infections, not everyone did. And so this is where the syndrome might have languished as a genomic disorder that was not very well characterized, except when I was a postdoctoral fellow, I discovered independently, I didn't know that this was a syndrome. We were looking for copy number variants or structural variants that were associated with schizophrenia. And I discovered that this same interval, the same 1.6 megabase deletion on chromosome 3 was associated with schizophrenia. And then later, there was some more data that was publicly available. And when I did a meta-analysis on all of the data that I could collect, I found that the 3q29 deletion is associated with a 40-fold increase in risk for schizophrenia. So this is a monster. This is an enormous risk for schizophrenia. For people who are familiar with this field, there's the 22q deletion. This beats the 22q deletion if it's an even higher risk factor. And so we think there's something about this deletion interval that is hitting sort of pathways that are relevant to schizophrenia and hitting them like a sledgehammer. And so this suddenly becomes a really important thing to study. And then it's sort of in an independent study that I wasn't involved in with a very large sample size, 40,000 subjects, looking for structural variants associated with schizophrenia, done by the PGC, the Psychiatric Genetics Consortium. These individuals were looking for structural variants associated with schizophrenia, and they found only 8 genome-wide significant loci, the 3q29 deletion being among them. So this is sort of this is an established schizophrenia risk locus, and we want to understand what it does. So 3q29 deletion syndrome, as I mentioned, associated with intellectual disability, autism, schizophrenia. It's present only in about 1 in 30,000 individuals, so it's very rare. About 20% of the time it's inherited, so 80% of the time it's de novo. 2 apparently healthy parents give birth to a child and later discover that there's been a mutation, and that child has 3q29 deletion syndrome. And so you guys probably know this, but we call this recurrent. When we see the same mutation happen over and over again, we call that a recurrent CNV locus. I was really interested in the syndrome because of the relationship with schizophrenia, and I sat down to read everything that there was about the syndrome, and I was done in about an afternoon. And I thought, well, I'm a researcher, and this is frustrating. Imagine if I were a parent of a child. It would be devastating to have a child with a genetic syndrome and have so little information. And I thought we could do better. And the first piece of that is to collect enough individuals, enough study subjects to actually do some work. And so I formed a registry, an online internet-based registry called the 3q29 Deletion Registry. It's housed at Emory University. We have now accumulated nearly 200 individuals with the deletion who have registered. And we have several ongoing projects devoted to understanding the phenotype, the neuroimaging profile, the molecular mechanism. We have a mouse model of the deletion, we have human cellular models, and so on. And we have an amazing team of collaborators. And we decided to call ourselves the Emory 3Q29 Project. And we even have a website and a logo and everything. So we feel very formal and very, very official. And things that we're interested in the Emory 3Q29 project, of course, are understanding the molecular mechanism, but also understanding the human phenotype, understanding the genetic drivers of the syndrome. There's a metabolic phenotype that we think is associated with the syndrome. And one of the things that we really want to understand is the genomic structure of the interval, what gives rise to the deletion. And there's a corresponding duplication that also arises, and we'd like to understand that too. And so, really, one of our primary questions is what causes the de novo event at the locus? So this is the interval. This is where all the money is. And so up here, we have an ideogram of chromosome 3, and that red box indicates, I forgot that there's one of these, and that red box indicates where we are on the chromosome. And I'm going to expand that region up. And here are the 21 genes in the interval. People who with 3q29 deletion syndrome have only one copy of these genes, and there's something about having only one copy that renders them uniquely susceptible to a host of neuropsychiatric and neurodevelopmental problems, among them schizophrenia. So this interval is flanked by 3 LCRs, or low copy repeats, and we're going to call them A, B, and C. The deletion we always see forming between LCRB and LCRC. People know what LCRs are. Everybody is familiar with that term. Should I say what it is? Say what it is. So low copy repeats. So everybody knows about ALUs and line elements, which are present throughout the genome in hundreds or thousands of copies. LCRs are present. They are not unique sequence, but they're present in a low copy number. There's another region of the genome, somewhere 1, 2, 4 places that bears homology. These two regions bear homology to one another. This LCR is 100 kb, and this LCR is 50 kb. And so when we think about NGS sequencing, short read sequencing, those pieces of DNA are 100 base pairs or 200 base pairs. And so we can't actually uniquely place them because they match perfectly to these LCRs. And so what we wind up doing is not sequencing those intervals very effectively with typical technologies. And so the reason why the LCRs are important is because there is homology between them. So it turns out that there's a segment within that LCRB that is homologous to a segment in that LCRC. And those homologous segments can align with one another and cause something that we call non-homologous recombination. And you guys probably know this. And that non-homologous recombination between those misaligned parts of the genome give rise to the deletion and the reciprocal duplication. And so the LCRs play a key role in the formation of the deletion. And the 3q29 deletion is not the only interval that is flanked by LCRs. Nearly all of the recurrent CNV loci that we know of, and these are just a few off the top of my head, they're all flanked by these LCRs. And so the LCRs are clearly a risk factor for causing this recurrent mutation. And in the 3q29 interval, if we look at a Muro-Peets figure, we can see that A and B, the LCRA and LCRB have homology to one another, and those are in an inverse orientation. And LCRs B and C have homology to one another, and those are in a direct orientation. At least this is what we know of based on the reference sequence. So we mentioned the LCRs are a risk factor one another, and there's homology between these risk factors. So they're probably, homology between these LCRs, and so they're likely playing a role in the formation of the deletion. But the other things that are interesting in the genome, the things that we are like these sort of silent, I think of them as sharks, right? There are sharks in the water, are inversions. Inversions are things that we can't see with almost all of the technologies that we have to interrogate the genome, but inversions probably also contribute to the formation of recurrent CNV loci. And so this is the reference allele, again, LCRA, LCRB, and LCRC, and this time the LCRs they're a little bit scaled. So this is really the size of the LCRBA in relation to LCRB and C. So an inversion has been reported between LCRs A and B. Recently that's been reported, that's about a 350 kb inversion. In theory, there could be an inversion between LCRs B and C, but that hasn't been published. No one has ever seen that as far as we know. And then there is a much bigger 1.9 megabase inversion that encompasses the entire region, and that also has been previously published. So we know that there are inversions in the region, and inversions have previously been shown to be risk factors for formation of CNV loci. So for example, this is an old paper. There's an inversion at the Williams syndrome locus. So the Williams syndrome locus is a deletion on chromosome 7q on 1.23. Williams syndrome is sort of a famous copy number variant, structural variant disorder, because individuals with Williams syndrome have what we call a cocktail party personality. They are very cheerful, they are very socially motivated, they really want to be your friend, and so there's something in this interval that's really interesting and that tells us a lot about social disability. And interestingly, individuals who have a duplication of this interval, who have the reciprocal event, have autism, or are more likely to have autism. And so some people have hypothesized that there's a dose response between the genetic dose in this locus and sort of sociability. And so in this study, which was published, granted, a long time ago, these individuals did FISH and they did pulse field gel electrophoresis in just 12 families. And they found that of parents where the de novo deletion formed, 4 out of 12 had an inversion. And this paper, which is not a lot of data, but a ton of effort, wound up in Nature Genetics. And then subsequent to that, some investigators did a lot more work. They interrogated 257 families, including 771 individuals, using metaphase and interphase FISH, which is a huge amount of work, and found that inversions were present in 25% of the transmitting parents, but only 6% of the non-transmitting parents. And so what that corresponds to is if you're someone who carries an inversion, you have a 5-fold increased risk of having a child with Williams syndrome. So that's super interesting, and it's telling us something about the formation of these structural variants. But it is a huge amount of work to do this. This is not high throughput. This is low throughput. And so, but clearly inversions are important, and we probably need a way to detect them. And other loci where inversions are hypothesized to contribute to genomic disorders include some of the recurrent CNV loci that we talked about previously. I mentioned the 22q deletion, which is a well-known deletion that contributes to schizophrenia. It's pretty frequent. It's probably 1 in 4,000 in the general population. And a few years ago, this paper popped up where these individuals assayed 17 families, and there were 34 parents of 22q probands using fiberfish, which again is a very laborious technique, very labor intensive. And they found that 16 of 17 parents, transmitting parents, were carrying an inversion. And so this seemed like further evidence. This is something out there, something we already know. It's very exciting. Inversions contribute to the formation of deletions, except it turns out that these investigators, this is how hard fiberfish is, it turns out that they'd misinterpreted their data and the paper wound up being retracted. And so this is like the worst thing that can happen to an investigator, right? And to the investigators' credit, as soon as they figured it out, they went ahead and admitted it and retracted the paper. But this points out how much of a problem we have in detecting inversions in the genome. So, okay, so we don't want to be subject to this, but we really want to interrogate LCRs and inversions at the 3q29 locus. And so what we need is a technology that's affordable, efficient. Yes?
Oh, no.
Oh, you didn't have a question? You're just agreeing with me, right?
Yeah, and actually the authors of the last paper, they published a beautiful paper just recently showing this exact locus and the complexity of it, using partially Bionano, but also a lot of other stuff.
Exactly, yeah.
They're still very keen on that locus and also correct their mistakes from the initial.
I think they did a great job. Actually, it turns out that I'm part of that consortium and I'm an author on that project. So yeah, but you're exactly right. You're exactly right. When you use the right technology, aspects of the interval become available. So if we want to do this, we need a technology that's affordable, efficient. It's got to be sufficiently rigorous so that we aren't susceptible to a retraction, and it has to be high throughput or more high throughput than typical technologies. And so we did some optical mapping at the 3q29 locus. So we performed Bionano Sapphire direct labeling whole genome mapping on 3q29 trio. So a trio is a proband who has the deletion and then 2 parents who don't. And then Bionano Genomics provided us some control data. So we wanted to ask about the frequency of inversions and LCRs and what the LCRs look like, and we wanted to compare that to people who didn't have a child with 3q29 deletion syndrome. And so we got some control data from Bionano Genomics. So the first thing that we noticed is that there's a reference sequence for LCRs, but we know the LCRs aren't very well characterized. And it turns out that in the LCRs in our parents, there is a ton of variability. And so this is an example here in LCR A. You can see that there's an expansion, and there's also an expansion here in LCR B. So there's lots of expansions, which is a surprise. And if we look at the frequency of the LCR expansions, you can see that actually almost no one has the reference sequence. That if we look at LCR, the reference allele is really the minority of the alleles in our sample of parents of a child with 3q29 deletion syndrome. That actually about 20% of our individuals have an expansion in LCRA, a bigger LCRA than is indicated by the reference sequence. 18% of our study subjects have an expansion in LCRB, and 35% have both of these alleles, have an expansion in LCRA and LCRB. Interestingly, we never see any expansions, and we don't see a lot of variation in LCRC. And so that's really exciting. It narrows down our search. It suggests that if the LCRs are contributing to the formation of the deletion, all the action is probably happening in LCRB and possibly a contribution of LCRA. Yep.
I mean, you have 200 normal controls for Bionano, so you don't really know how common this is in the general population to have these expansions, right?
This is true. This is true. Right. So we haven't actually compared our data yet to Bionano's data. We've done that for inversions, but not for expansions of the LCRs. But you're exactly right. And that's exactly right. That's the question that we have to answer is, what does the general population really look like, right? I mean, our data suggests that the reference allele is not really the allele in the population. So, right. You're exactly right. Other comments or questions? Okay. So the other thing we can do here is we can detect inversions. And so this is what an inversion looks like. These markers are all reversed in orientation compared to the reference allele. And this particular parent is heterozygous. They have one reference allele, and they have one allele that's inverted for this 350 kb segment that is just upstream of the 3q29 deletion. And so now we do have frequencies of inversions in the control population. And we ask about that. It turns out that the frequency of this AB inversion in our parents is very different from the general population. About 14% of our parents have this AB inversion, but only 2% of controls have this inversion. Now, it turns out that the data that we have may not be ideal because they were done, I think, with the iris technology. And so there may be limited resolution of this. And so we currently have better data and are in the process of interrogating this. But right away, this suggests that maybe inversions are related to the formation of the deletion. And interestingly, the inversion that is sort of present at a higher frequency is upstream of the actual deletion interval. And so this is one possible configuration. Perhaps an inversion between LCRs A and B interferes with pairing of the chromosome and leads to, and the machinery is looking for homology and doesn't find it until they get to LCRs B and C, maybe. So this is exciting data. It's early days. Yep.
Can you check whether it's transmitting parent or not?
Yes. So that's why I asked this question about parent of origin, because we really need to figure that out so we can, we need to know who's the transmitting parent and who's not.
It should be relatively simple, so that the deletion in between is relatively large. I assume there is some common SVs already in that region itself. So
not that we've been able to find, unfortunately. Yeah. So and we tried to use, so there's a labeling pattern, and we wondered if we could just extract that labeling pattern, and it turns out that it's not quite as accessible as we would have hoped. And so we're still working on that, but it's so much labor. In the end, we were like, we're stupid. We should, we have data, it's exciting. We should just run a SNP, it's so easy to run a SNP array. But now that we have the SNP array and we know which parent is transmitting, we can go back and ask if there are ways that we can extract data to tell us that information. And to the Bionano people, we would love to work with you on this to figure this out. So anyway, but right, so that's one thing that we really need to know. We've looked at our parents as a group, but we could split them up into transmitting parent and untransmitting parent, and that's going to be non-transmitting parent, and that's going to be really important. Okay, so all of this is really exciting about the formation of the deletion, but we discovered something that was really surprising to us. The thing that we discovered is, so we get, the way that we recruit study subjects to our registry is we ask, do you have 3q29 deletion syndrome? And they all say yes, and they furnish a clinical genetics report. And the clinical genetics report always has the breakpoints at the same point, which are kind of around the LCRs. To our surprise, when we processed our trios using Bionano, we discovered that the breakpoints are actually different. They're all different in our different probands. Some of our probands have the same breakpoint, but particularly in this LCRC, there's lots of different breakpoints. And what was even more surprising to us, so now we're going to zoom in on the centromere oxide. And on the centromere oxide, this is LCRB. This is where we have hypothesized. We've said this is the canonical 3q29 deletion. And you can see that these breakpoints are all different. And maybe that's not a surprise, but all the breakpoints are contained within the LCR. When we look on the other end, though, to our surprise, on the telomeric side, the breakpoints are really different. And there's a gene outside on the other side of the 3q29 interval. And it looks like, for at least some of our study subjects, that gene appears to be deleted, appears to be partially deleted, and is potentially part of the syndrome. And we've gone in our study subjects and asked about expression of this new gene, RUBCN. And it turns out that for these individuals, RUBCN is in fact has decreased expression. And so all along, we've been thinking about 21 genes as part of 3q29 deletion syndrome, but there may be a 22nd gene. I've also talked about how there's heterogeneity in the phenotype. I wonder if some of the heterogeneity in the phenotype is because some of our individuals have another gene that is deleted. And so this is an area of very intense investigation for us. But I think it speaks to some of the limitations that can exist in SNP-based clinical microarrays and how Bionano technology could potentially help us uncover some of that work.
Can you comment on the discordance between those two results? Because at the end of the day, SNPs should be able to see the variations unless while they're reporting the flagging regions rather than the containing regions.
So you're totally right if the region was in unique sequence. The problem is that this is not unique sequence, and because it's not unique sequence, we can't really synthesize oligos that tell us very much in the interval. And in fact, if you were to plot the SNPs on if you were to plot by chromosome length or by chromosome position, the SNPs on a microarray, there would be a big desert, a big blank spot that corresponds to these LCRs because we just can't put SNPs down in them. So you're totally right, but the LCRs ruin our lives. So, in conclusion, we suspect that inversions may be associated with formation of the 3q29 deletion. There are also expansions in the LCRs that we didn't know about, and these also may contribute. Expansions that aren't captured by the reference genome that may also contribute to formation of the deletion. And we wonder if there are some interactions. We wonder if there's some configuration of inversion plus LCR expansion that really leads to someone having a very high probability of developing a de novo event. And to our surprise, there may be a 22nd gene that's contributing to phenotypes in the interval, this gene RUBCN. And so all of these things would not have been accessible to us without optical mapping technology. And so we're really learning new things all the time. I want to thank my team. I have an amazing team. They're incredible. All of these people are involved in the 3q29 project. This is a project of passion for us. And so I'm very lucky to have such great collaborators. And this is a slide that Bionano wants me to tell you, wants me to show you. So thank you very much for listening. I'll be happy to take questions.